In this lecture, I'll be going to talk about monoclonal antibody. Mono means one and clonal means clone. So when antibody is producing from a single clone of B cells, it is called monoclonal antibody. So mono means one or single, clonal means clone. What is the use of this monoclonal antibody? There is a therapeutic use and research purpose. Mainly these two functions is performed by monoclonal antibody. Produce a huge number of antibody. We cannot just depend on a B cells. We have need a immortal B cells. So let's assume if I want to produce a monoclonal antibody against a antigen like antigen X, what I have to do? I have to inject this to a mice. Then after a few days, I will separate from the mice a spleen and collect anti-X antibody producing B cells. Okay, as I have mentioned that monoclonal antibody requires a huge amount because it is actually requires for the therapy and research, so it has an industry. So we need a cell that is immortal. Fuse this B cell with another B cell called myeloma cell. What is myeloma cell? I am telling you. A B cell, when activated, it is called plasma cells. Plasma cell means when a B cell can produce soluble antibody, it is called plasma cells. But when this plasma cell become cancerous, it is called plasma cytoma. When this plasma cell remain in a single bone, this is called plasma cytoma. But when this plasma cytoma starts to metastasize, this is called metastasis, starts to metastasis into multiple bone, this is called, the cell now is called multiple myeloma or simply myeloma. So now you know what is myeloma cells. Myeloma cell is nothing but a tumor or cancerous plasma cells, or you can say another way, it is an immortal cell. So to produce an monoclonal antibody, huge amount, we need a immortal cell. So we have chosen this myeloma cell and we have to fuse these cells with the anti-X producing B cells. For the fusion, we use a chemical called PEG, means polyethylene glycol, PEG. This chemical helps to fuse cells. After fusion, what happens? Some cell become fused. It is B cell and this is myeloma cells. But some cells remain not fused in conditions. To only select those fused cells, they have to go through a selection process. So we have to select only this cell and have to remove the other cells. So scientists have used a very innovative technique. What they have performed, so after this, we have to select only fused cells that are called hybridoma. So we have to select these hybridoma cells. So what is the procedure to select these hybridoma cells? Is using a medium called HAT medium. So for the selection, we have to use a HAT medium. Hypoxanthine, this A for aminopterin and T for thymidine. So these cells have to go through a selection process in which these three chemicals are present. So actually, scientists have taken the advantage of purine biosynthesis. They have taken the advantage of purine biosynthesis. So I am talking about the selection method. So this selection is very important. Purine biosynthesis can be performed in two ways. One is called de novo, from the beginning. De novo means from the beginning, and another is called salvage. So purine can be synthesized in this way, de novo pathway and salvage pathway. For the de novo pathway, one important chemical is called tetrahydrofolate is required. So for de novo pathway, tetrahydrofolate chemical is required, and for the salvage pathway, one chemical is required called HGPRT. It is the enzyme. The full form of this HGPRT means hypoxanthine guanine phosphoribosine transferase. You don't have to remember the name, but you just remember the HGPRT. And we are using a medium called HAT medium, in which there is hypoxanthine thymidine and aminopterin. But actually reason of using this medium, aminopterin is a chemical that can inhibit this tetrahydrofolate. Okay, so this medium will enable every cell, it may be B cell, it may be hybridoma cell, or it may be myeloma cells. These 3 B cells, there is actually 3 possibility, one B cell or one hybridoma cell or one myeloma cells. If these 2 cells not fuse, they remain as B cell, myeloma cell, and if they fuse, they become hybridoma cell. So there are 3 possibilities, but these 3 cells will not go through de novo pathway because here in the medium one enzyme is present called aminopterin that actually inhibits tetrahydrofolate, and tetrahydrofolate is required for de novo pathway. So these 3 cells, B1 or hybridoma, B cell, hybridoma, and myeloma cell will not go through de novo pathway. De novo pathway is totally prohibited for these cells to replicate, because as we know that purine is important, purine is a nucleotide, is important for replication. So if one cell do not replicate, it will die. So only the pathway is open, that is salvage pathway. Only one pathway is available. So both these cells can go through salvage pathway. Okay, the next important thing is, as they have chosen these myeloma cells, there are 2 conditions for the selection of myeloma cells. One, the myeloma cells do not produce antibody. So there are 2 conditions for the myeloma cell to select. As I have already mentioned, this myeloma cell is actually plasma cell, cancerous plasma cell. So the plasma cell can secrete antibody. Select this myeloma cell in this monoclonal antibody generation. The myeloma cell do not produce antibody, because if this myeloma cell is producing antibody, and if this antibody, anti-X antibody producing B cells are also producing antibody, then the 2 antibody will mix, and there is very difficult to separate these 2 antibody. So to avoid this situation, we have to manipulate the myeloma cells that the myeloma cell will not produce or do not produce antibody. So first condition for myeloma cell is it will not produce antibody. And the second important fact is this myeloma cell lack HGPRT enzyme. Now you understand. So the myeloma cell will not replicate in this medium. So the myeloma cell will be degenerated. So the myeloma cell cannot go through salvage pathway. But now this hybridoma cell, as these hybridoma cells contain B cells and these B cells contain HGPRT, so this hybridoma cell can be survived and this B cell can be survived. But there is no problem with these B cells because this B cell is producing anti-X antibody, but eventually these B cells will die because it is not immortal. But there is no problem because there is hybridoma cells.